Guys, welcome. We are doing our next block of material, protein interactions. And I wanted to start out with this kind of landscape view. From the genome, there is information that can be gathered. And eventually, coming out of that information are hypotheses, questions that then can be tested. Okay, that's what science is all about. So the informatics side is not just to gather data, it has to be useful data, it has to be integrated data. So this is showing some of the different parts of that integration. Some will be found in molecular biology. Okay, I'm just going to kind of circle those. Okay. And then the others I'll underline will be in this lecture or we've covered. We've covered protein localization within, this is a worm, an organism, okay? And we're going to hit then protein interaction to understand protein X and Y come together. What do they do? Where are they doing it? Who are they interacting with? You know, what's the order of interaction? Do they show up in a disease or not? These are some questions. So there's, you know, kind of a grand hypothesis, but each technique does have questions as well, and those, you know, should be pursued. So protein interactions, PPIS, we like to give names here. This is the basis for how structure and function of a cell, understanding cellular physiology, is built. Proteins are not doing an activity by themselves. They're part of a team. If you think of a sports team, they're all part of a team inside of a cell. And so finding interaction partners leads to understanding function that then can be used for, you know, applied purposes such as therapy. Okay, so this is kind of a introduction to PPIS. Here's a paper, Imaging in Biological Research, and the interactions that we will be trying to find, the subtext that they're in is cellular function. Okay. Important cell functions are how cells cycle, divide, go through G1S, G2 mitosis. How cells receive and transmit signals, right? That's, as an organism, our different organs secrete hormones, other bioactive proteins that then change the status quo. And so cells that receive that signal, that process is known as signal transduction. A hormone comes and is released. What does it do, right? If you eat a sugary meal, then your pancreas needs to break down that sugar. You have glycolysis and the beginning of food production or energy production from food. Well, insulin is released by signal transduction. Okay. And then metabolic pathways. Metabolic pathways. These are activities that a cell does inside a tissue or an organ, and that's what make that tissue or organ. That's its big function. So in the big organismal scale, these interactions that we're going to study regulate signals that affect the body's homeostasis. If we're developing fetus, we just went uh, fertilization, then different cells had to morph into differentiated cells, and you get a pattern of development, right? You have normal physiology, and then of course when things are not right, we get disease in living animals. And a lot of times the protein-protein interactions are different. There's too much interaction, not enough interaction, these kind of things. Anything off the normal balance. Obviously, therapeutic targets are how we can use this protein-protein interaction. The pathways that proteins have in tissues do produce regional effects. It's hard to do this in a test tube. So we're kind of making our way back. We did cell culture because we couldn't really mimic the animal, and now we take that information from an in vitro setting and go back in vivo to try to apply what we've learned to try to, use that as therapy for certain diseases. So here's another paper. Molecular determinants of protein oligomerization in neurodegenerative disorders. And it looks like they've got a technique, I'm not sure we'll cover it, but biomolecular fluorescence complementation. And I just like their introduction because they speak about protein-protein interactions here. And I think most biologists realize that's the root of all biological processes in both normal physiology and disease or pathologic conditions. And so our ability to study those did rely on protein biochemical assays. And we're going to talk a little bit about immunoprecipitation. This is immunoprecipitant. So this is an antibody that pulls a protein out. Immunochemistry, detecting proteins in a histological slide in a part of a tissue or cells using antibodies, and you visualize it by cytochemistry and you see where it's located. You can do assays for a protein's activity in cell lines, cell cultures on cells, tissue lysis, fixed cells, or tissue themselves. So these assays certainly have moved us to this point. They're very informative. They can be accurate and widely used. But again, do they have the study to get real time? And that's where we're going with some of this, is real-time protein interactions in living cells and organisms under the conditions. And I think the reporter methods are coming along. They will be using nanoparticles and other kinds of reporters. They're already putting recording devices inside humans to figure out what's happening in real time. Eventually your doctor can monitor you by GPS, right? How are you doing? It's coming. I don't know when, but it's coming. Real time in living cells right now, it's still, they hadn't figured it all out. A lot of labor, expensive, time consuming. So what we've done the last 20 years has led up to trying to characterize, and we all have words here, interactome. So every protein has an interactome. Okay, this is all the proteins that it can interact with. These can be during development in fetal, early adult, after puberty, old age, Proteins can, let's call it handshake, at various times in the cell cycle under certain stimulation conditions. Uh, and we would love to visualize these interactions in living cells and certainly carry out high throughput genetic and pharmacological screens for modifiers of these protein interactions. So do things like our diet, do things like chemicals in the environment, do they modify our protein-protein interactions? Is that part of a long-term issue we need to be aware of? So besides the biochemical assays and precipitations, and there's new techniques that involve fluorescence, bioluminescence, and protein complementation. And so this is kind of, we're trying to move from cell lines and in vitro kind of back to the animal. So we've kind of come full circle and just kind of realize that's where we're going back. Now realize that synthetic biologists may be a way to figure this out in a different way, actually creating organisms that can be highly viewed and all the information gathered from a synthetic organism to mimic what certain cells, tissues, and things occur. So that's kind of on the horizon as well. But we have an interactome. So every protein has an interactome. So protein-protein interactions are physical and selective contacts that happens between a pair of proteins. These could be domain or regions, motifs. So they just call it certain molecular regions. So you could think of a domain or even a motif inside the domain, and there's a context. Interactome is the totality of all those interactions in a cell, organism, in a context, a disease. So they use the word interactome to interact or to designate at the one protein level, but the definition can extend to everything happening in a certain type of cell. And so these interactome or interaction networks, okay, can be graphed and figured out, and there are graphical representations in which proteins are nodes and their interactions, who they are their edges. So every protein is a node that has edges, and these are kind of the graphical maps. This allows for a big visualization of disease, of a situation here. So why protein-protein interactions? Here's the central dogma. I don't know what happened there. This is my kind. Let's see. I don't know what happened there. My protein-protein interactions, central dogma, DNA, RNA, the protein, and then one protein, one function here. And that's not true anymore, if it ever was true. A protein has an interactome. It has end functions. We may not know all the functions, and it participates in unknown networks, and that's what we want to define. There's the interactome here. And so they're showing the handshaking, and again, there can be predictions here. And notice this protein interacts with that protein through another protein, okay? And so think about how you regulate these interactions, and they're just showing these graphical representations of a protein and its interactions there. Let's see. So to predict a protein's biological function, you try to figure out who they're associated with. So you might guilt by association. And the idea is that proteins with similar functions should cluster together here. Should cluster together. So people try to interact or draw a draft map which puts all the processes and pathways into the map. That map can include theoretical pathways defined through informatics, wet lab. It could be tying in all the medical papers and knowledge that have been published about a disease or a situation and put that into the map. So if you think about these maps, these interactome graphical displays here, there's a lot of data that's feeding in. And sometimes it's just theoretical. And you go with the theoretical until something changes it. So just do be aware some of these are interacting. So here's a protein. The main protein here is CCNT1 in green. It's a cyclin protein. It regulates a kinase. A kinase is an enzyme that adds a phosphorus group. Usually when you add a phosphorus group to a protein, that adds a level of energy and that activates it. CCNT1 can regulate genes coming on and off, transcriptional regulation. And since it's in the family or has a annotation of a cyclin, it's involved in regulating how our cells divide here. And so all around are multiple proteins in CCNT1's interactome. So for instance, this one here only is around in early development or involved in making new blood cells. So very limited. Here's a protein, a proto-oncogene transcription factor that CCNT1 interacts with. Here is a kinase that it interacts with, CDK9. Here's another transcriptional regulator. SMAD is a modification. SMAD1, SMAD3. Here's transcriptional activations here. Notice some of these are oncoproteins. So, and the CCN1, if it misregulates these proteins, can cause cancer, because that would probably mean that genes are on when they shouldn't be on, or they're off when they shouldn't be off. So this is when they discuss guilt by association. These are the predicted proteins, or they've been wet labbed to know that this interacts with that. So this protein is a node, and these are its interactomes. So you would predict there may be others. It's kind of the unknown unknown, and you would predict that any new proteins that are involved here will be involved in transcription or cell cycle control. It would be very surprising if it was involved in, say, mitochondria, okay, or Golgi function. That would be completely different from who it's been interacting with. Very surprising. So probably that doesn't happen. So here's a more advanced characterizations here of different pathways. Not you have to memorize that, but in these software, they actually use colors and sometimes even thicknesses of lines to define subgroups in the interactome. And then they build these pathways. So here is a core unit here. I'm not sure, proteatic to proteasome. These are proteins that chew up other proteins. And looks like you have 14 different proteins that make up this proteasome. It's called proteasome. And then they have 20 proteins that are regulating it on either side. Okay, so this is heavily regulated, which makes sense. You don't want to start chewing our proteins up before there's a signal for apoptosis or other kinds of modifications. Okay, so protein-protein interaction can be predicted by wet lab work, and this is some examples. We will talk about the yeast 2 hybrid Y2H. Okay, this is a cartoon showing that. Tandem affinity, it combines different affinity tags and then a mass spec analyzation of these mass charge to figure out these proteins involved. And here's a protein attached to an affinity column, and then you do this under conditions where its partners interact. You can then break these partners up and analyze them by mass spec. So that's what's going on here. So all these different proteins are being analyzed. So you can take any protein and figure that out at that snapshot in time. So that's in a certain cell type, a certain disease condition, and you could test this interaction under other conditions. Will you bind all 4 of these or not? Maybe it doesn't bind any under certain conditions. It's just good to know, and this data is collected here. Proteins can be made into a crystal. It's frozen in space in a 3-dimensional way. And then you can hit with X-ray, and it scatters, so it's kind of like hit and all the bullet fragments scatter. Well, all these X-ray fragments scatter, and you can figure out the scatter, and that can be translated into a structural shape where all amino acids are sitting in a frozen condition. And then you can make a model here, and this is a multi-protein complex here that they crystallized, and now they define the different proteins under different conditions, and you end up getting a structural model that way. So these just tell you the protein interacts, and we'll talk more about that. This actually starts making predictions to domains or areas where these interactions are occurring. So I say this in molecular, and I'll say this here. How do you prove, say, this area of the CUL1, how do you prove that area is in binding the blue protein? Okay, you can mutate it, get rid of it, delete it, and therefore it would not bind. Therefore, the crystallization pattern would be different. You wouldn't have this kind of these things binding to it. So you can do a mutant version of a protein and compare it to wild type, right? So you can do disease versus non-disease and compare its structure as well. So you can compare what proteins bind, wild type and disease, and gives you insight. And there's just, this slide is just going through, and we'll talk, the reason I'm not defining the 2 hybrid yet, because we're going into that. And the TAP, tandem affinity purification, you've seen mass spec, you've seen affinity, and this is a combination of the 2, and then the protein crystallization, we'll talk about that. But no single method can produce a true binary interaction under physiological conditions. So anytime you pull a protein out and you crystallize it, you're taking it out of the cell. They want to determine, does the proteins really interact? Okay, it takes multiple methods and predictions to be confident about what here. So if you have binary interactions, you have 2 proteins binding. You can do co-localization. Two proteins are very close together in the cell. That's suspicious. You can do the tandem and find a complex purification, so you can find n number of interactions here. And a functional direct interactions like an enzyme and substrate are inhibitors that inhibit enzymes, so you can actually delve into that. And so binary interactions, two proteins coming together. We'll talk about the yeast 2 hybrid in more detail. This is just a diagram here, Y2H. And this is just showing one of these diagrams of an interactome here. And this is looking at fluorescence here and adding a ligand to an enzyme, and now they're interacting, and you can detect that interaction by physical biochemical methods. And then here's co-localization. Here's multiple proteins together. So here's actin, vinculin, and when you in ovary cells, and when you take the two fluorescent patterns and you overlap them, red and green make what? Yellow. So now all the yellow areas of the ovary cells are the areas where there's both actin and vinculin together. Green is actin only, and I don't know if there's any, vinculin only, and I don't know how much red only there is. So it looks like there's more vinculin that interacts with actin, but not more actin that interacts with vinculin based on this figure here. Okay, based on that. And so how many proteins are interacting here under different conditions? So here's just showing an affinity tap where you've actually gathered all these subunits together. Here's the crowd here. And when we talk about going inside a mitochondria, being imported in, there's a lot of machinery. The TIM and the TOM complex, we'll talk about that. There's the TIM and the TOM. These are a complex of proteins that they found together that works to bring a protein in, assuming the protein is the right zip code. Okay, and there are other proteins involved, multiple ways to move into the intermediate space, inner mitochondria membrane, or even into the matrix. So in a mitochondria, a protein could stay on the surface, can go through the intermediate space, can go through the inner membrane, or end up in the matrix. So 4 different locations. So there's probably protein information when it's taken in. Where does it go? So if you had to study a mitochondria protein, where does it end up in the mitochondria? How would you predict that? Okay. This is direct functional interactions, usually in the test tube. You usually have a good, strong evidence that the proteins are identified and they're known. So we kind of predetermine. Okay. So if you have an enzyme, there's usually a substrate, and this is a way that you can, like I said, purify and crystallize a protein, find its structure, a lot of alpha helices there. And then here's a model of the enzyme and the substrate complex here. And you can figure out the different interactions, and you have a detector looking at the different interactions under different conditions. You can see the enzyme and substrate separate, you can see them together, and you can see the product and the enzyme back. Okay, you can detect those. So these areas in little kind of lines here are the known motifs or amino acids that handshake. Notice that there are amino acids here and there. So when we talk about an interacting, it takes two parts of the protein to interact. So again, you can study this side or that side. You can study both. And here they are. Protein 1, also known as protomer, protomer 2, and then this is where they interact. And this is a model showing those strong interactions. So you can imagine looking at one's genome, make predictions, eventually they will, on how strong our interactions are. Imagine you were born and there's only, say, 5 interactions instead of the needed, 7 or 6. That could be a problem. Okay, so that's where they're going with the genome project on that side, is to look at all the predicted interactomes and then look at individuals and see whose interactome is very good or not so good for the interactions there of the multiple proteins. And it just shows the overlap in sequences looking at the areas here. So they're just defining those areas that have the interaction in the genes. Okay, so if you were born with a deletion here, then this yellow protein wouldn't have this region, and so this would never interact with these two proteins. So there are complexes, but how do those complexes interact together? Okay, we can make models and we can get information, the affinity purification technique, but there may be more different structures, and it's just showing different potential interactions, 15 versus 5, but in reality it may look like that. So we're just in the beginning of trying to solve using theoretical algorithms to make predictions about these interactomes, especially defining complexes, okay, and figuring out how they actually bind together. So there's a bunch of databases out there. So if you had proteins, you could look at all these, and this is probably not up to date, but here it is. I know one group's found string, okay, but here are some other ones, okay, and some are shallow in the amount of data and some are very deep. So it just shows you the different levels here. Okay, so we are stopping here and we will begin with an introduction into interactions, and we'll go from there on yeast 2 hybrid and the other methods, wet lab methods anyway. Okay, let's see if I can get out of here now.